afternoon, students. I am Dr. Sanskruti from Department of Biotechnology. This time we will discuss about a topic from your cell biology course, that is cell signaling. In the earlier session, we had discussed about cell transport. So the cell signaling topic is in continuation with your cell transport lecture. So here, if you can remember, in the earlier session, we had discussed about different transport. There are different types of proteins useful for transport, that is, carrier mediated transport or channel proteins are useful for transport. And then later on, we had seen that based on the energy required, if the energy is required for transport, it is known as active transport, and if energy is not required, it is known as passive transport. And then later on, we have moved on to study about the basics of transport, that is, we had discussed about the bulk transport, where we had discussed about phagocytosis and later on about the pinocytosis. Now, in today's session, what we'll do, we'll try to connect the transport with cell signaling and see how the transport mechanism is also useful for cell signaling. So, in today's session, first of all, we'll discuss what is cell signaling. That is, we'll discuss about the mechanism of signaling. That is, how the signaling takes place, how the signaling molecules are released, and how they bring about the cell response. Then we'll see about different forms of signaling based on the different signaling molecules used in signaling and where they go and bind. And later on, we'll see how the receptor mediated cell signaling takes place. So let us first see what is cell signaling. So cell signaling is required for cells to communicate with each other. So as we human beings to live in the society need to communicate with each other. So this form of communication is mostly verbal, written, or in the form of action. So why do we require communication with the members of our society, that is our neighbors, relatives, or friends? That is for our development and existence. Similarly, cells need to communicate with each other, that is with the neighboring cells, to bring about their differentiation, cell differentiation, then division, and even useful in development. And further, not only development, growth, and division, but also in differentiation, migration, and apoptosis. So, how the signaling takes place? So, the signaling takes place by the use of some chemicals known as chemical signals. So basically, chemical molecules are useful for signal. They can be small molecules like hormones, some proteins or enzymes are used in cell signaling. So based on what type of proteins are used or what type of molecules are used, there are different types of signaling and there are different mechanism of signaling. And even based on what part of the cell the signal goes and bind and what kind of response is generated, there are different types of signaling. So moving on before to the different types of signaling, let us first discuss the basic mechanism of cell signaling. So as I told you that the signaling molecule is required for signaling. So this extracellular signaling molecule, that is the molecule released by one cell, goes and bind to the receptor present on the plasma membrane of another cell and further bring about cell response. So what happens in cell response? Some intracellular signaling molecules are activated. As you can see in the figure, when the extracellular signaling molecules binds to the receptor, it activates the cell signaling cascade. That is, the intracellular signaling proteins are activated, which finally go and activate the effector proteins. So the effector proteins or the effector part of the cell can be different. So if your signaling molecules go and bind to the metabolic enzyme, as we know, metabolic enzyme bring about changes in the metabolic processes, like different metabolic cycles. So metabolic processes changes will be done or regulations will be done when a signal goes and bind to the gene regulatory proteins, that is the proteins involved in gene manipulation. So here the genetic level changes will be taking place in the cell. And when the signaling molecules goes and bind to your cytoskeletal proteins, and as we know that cytoskeletal proteins are required for effective support to our body or support to the cells. So there can be conformational changes taking place in cytoskeletal proteins as per required by the body by this external signal. So this is the basic mechanism of cell signaling, where it is important that the extracellular signaling molecules is released from the one cell and it goes and bind to the another cell, or it can directly transfer from the cell membrane of the another cell. So let us see how the signaling molecules bind to the cell. There are two ways by which the signaling molecules can bind to the cell. First, as we had discussed in the earlier session in the transport, that the molecules can be hydrophobic in nature as well as hydrophilic in nature. So when a molecule is hydrophilic in nature, what does it mean? So hydrophilic molecule is a molecule which requires water or which loves water. So usually our cell membrane is lipophilic in nature, that is, it is made up of lipids. So this molecules which are hydrophilic in nature cannot pass through this lipid molecule. So they need to bind to the cell surface receptor. So as you can see in the figure in the A mechanism where the hydrophilic signaling molecule is there, it goes and bind to the receptor present on the cell surface and then bring about the cellular response. Whereas in the another case, that is intracellular signaling, or when intracellular receptors are there, and if molecule is hydrophobic in nature, that is molecule does not dissolve in water, that time it can directly pass through your cell membrane. So molecule can directly diffuse through your small cell membrane, that is usually which is smaller in size, that is small hydrophobic signaling molecule will diffuse through your cell membrane and bring about cell signaling. So these are two mechanisms by which the cell signaling takes place, that is your signaling molecule released by one cell goes and bind to the receptor present on another cell and by directly diffusing and bringing about the cellular response. Here in the second case, as I say, intracellular receptors, that is the signaling molecules go and bind to the receptor present inside the cell and bring about cell response. So these are two mechanisms of cell signaling. Now let us see how there is a different forms of signaling based on the how the signaling molecule is released. So I told you that the signaling molecule can be any molecule which is in small in size, can be hormones, proteins or enzyme. So when the signaling molecules are released and they go and bind to the different parts of the cell or different cells is based on where this targeted cell or the cell where the signaling molecule has to bind is present, whether it is present in the near vicinity of the cell which releases a signal or is present far away from that cell. There is two type of signaling, that is short or long distance signaling. So this signaling is divided basically in three forms: autocrine signaling, paracrine signaling and endocrine signaling. So let us discuss each type of signaling in detail. First, about autocrine signaling. So this kind of signaling is self-signaling. So what happens here? The signaling molecule released by the cell binds to the receptor present on the cell itself. So here, if you can see that there are different molecules which are used in signaling. So in this type of signaling, hormones or chemical messengers released by cell goes and bind to the receptor present on the cell itself. So you can see in the image that the signaling molecules which are released, the red color signaling molecules which are released by the cell goes and bind to the receptor, the green receptor present on the cell itself. So you may wonder why the cell has to bind to the receptor or the signaling molecule released by the cell itself. So this takes place when the cell has already differentiated, that is the cell division has taken place, cell is distributed and is differentiated. Now this type of signaling molecules are helpful for the cell to have the decisions regarding the development. So you know that there different kind of development take place in a different type of cell and they play a different role in our body. So such kind of roles is destined on what signaling molecule is released by the cell. Here I would take a better example of cytokine interleukin 1 in monocytes. So monocytes usually release this type of interleukins for having an effect or showing an immunographic effect, that is mostly immunologic effect, and even towards the allergens this effect is shown by the signaling molecules. So here in the autocrine signaling, the high concentration of the signal is secreted, that is the signaling molecule which is released by the cell is in very high concentration and goes and bind to the receptor present on the cell itself. So autocrine signaling is called as signaling by cell. Then there is another type of signaling known as paracrine signaling. So earlier the signaling was, that is the signaling molecule was binding to the cell itself. Now in the paracrine signaling, the signaling molecule goes and binds to the cells presented in the proximity, that is the target cells are present in the neighboring vicinity of the cell which releases the signaling molecule. So these signaling molecules are rapidly taken up by the neighboring cells. Why rapidly taken up? Because there are some kind of enzyme, extracellular enzymes or some other molecules present near the vicinity of the cells which can degrade the signal or weaken the signal. So this type of signal is rapidly taken by the neighboring cells. So here you can see in the image there is a central cell from where the signaling molecule is released and it goes and binds to the receptor present on the targeted cell. So in this kind of signaling, the receptor plays an important role to bind the signaling molecule. Your example is neurotransmitters. If you know that best example of neurotransmitter which work in our body for nerve impulses, acetylcholine. So acetylcholine, if you can remember, is acetylcholine released from one cell, that is the one part of the nerve cell, goes and is released into the synaptic cleft of the cell, and it is, as soon as it is released in the synaptic cleft of the cell, it is taken up by the neighboring cell where the receptor for neurotransmitter, that is acetylcholine, is present, and it finally brings about the nerve impulse or nerve signals. So this is the best example of paracrine signaling, where the signaling molecules goes and bind rapidly to the neighboring cell. The third type of signaling is endocrine signaling. So you know that endocrine cells are there in our body which are secreted from the endocrine glands, and this type of cells usually secrete any molecules directly into the bloodstream. And there's an opposite kind of cells similar to endocrine cells, or opposite to endocrine cells are exocrine cells. So as the name suggests, what these cells do? These cells directly synthesize or directly release the molecule on the surface of cell or on the surface of epithelial cells, whereas endocrine signals or endocrine cells release the signals directly into the bloodstream. So this type of signaling is required where the cell is present at a distance, the targeted cell is present at a far distance. So here what will happen? The signaling molecule released by the endocrine cell into the bloodstream will go and bind to the signaling molecule, or go and bind to the cell which is present at a distance. So here also again it goes and bind to the receptor of the targeted cell. So this type of signaling is useful for distance target cells, or the cells which are present away from the site from where the your molecule is released, that is the signaling molecule is released. So here we have seen that based on the distance and based on where the molecule goes and bind, there are three types of signaling: autocrine signaling, paracrine signaling, and endocrine signaling. Now, here in the endocrine signaling also, I forgot to mention the example. The example of endocrine signaling is again insulin. So here insulin, you know, that is directly released by the beta Langerhans cells into the bloodstream, and it goes and directly binds to the receptor cells or receptors of the targeted cells. So here we had discussed different types of forms of signaling based on where the signaling molecules goes and bind. Now there's another type of signaling mechanism, that is receptor mediated signaling. So in all the signalings we had seen that in the earlier signalings that the signaling molecule was going and binding to the receptors present on the targeted cells. So what are these receptors? So receptors are the molecules present on the cell surface which help the signaling molecules to bind to the cell. So as we had already studied, there are different kinds of signaling molecules, hydrophobic or hydrophilic molecules. So hydrophilic molecules cannot directly pass through the cell membrane via the help of some carrier proteins or the channel proteins. So they have to bind to the some kind of receptor. So there are three types of receptors through which the signaling molecules can bind. They are G protein link receptors, ion channel link receptors, and enzyme link receptors. So today, now after this, we'll discuss each type of receptors in detail. So first, let us discuss about G protein link receptors. So G protein link receptor, as the word suggests, it is this type of signaling or this type of receptors are linked to G protein. So what is this G protein? So G protein is a 7 transmembrane protein. As you can see in the image, it has 7 different domains. The orange color protein which is present on the cell membrane, it has 7 domains and it passes 7 times from the cell membrane. So it is known as transmembrane protein, and the structure of each this 7 domain is similar. So it is known as homologous or homologous proteins. And this G protein is basically linked to GDP or GTP. So GDP is guanosine diphosphate. When this G proteins are in inactive form, that is when they are not working, that time they are present in GDP form, and when they become activated by some kind of signaling molecules, they convert to GTP and then bring about cell response. So let us see how this response works. So when an extracellular molecule, that is your ligand or your signaling molecules, goes and bind to the extracellular part of this receptors, that is the outer part of this receptors, there is an activation of this GTP or activation of G proteins. So what happens in activation? In activation, GDP is converted to GTP, and after conversion of GDP to GTP, as you can see in the image, this GTP protein is made up of 3 subunits, alpha, beta, and gamma. And after your phosphorylation, that is conversion of GDP to GTP, these two parts get separated, that is alpha subunit and beta and gamma subunit get separated, and that can further go and bind to another intracellular molecule or intracellular receptors and activate them to bring about cell response. So this is how G protein linked cell response work. So let us now discuss the second type of signaling, that is your ion channel linked signaling. So as the word suggests here, ion channels are responsible for uh, activating the, or ions are responsible for activating the this kind of receptors. So if you can remember, in the transport we had already seen there are different types of ion channel depending on the ion gated channels, depending on what type of ions are used. For different ions like calcium, magnesium or zinc, there are different channels working. So they are known as transmitter gated ion channels. They are involved in rapid synaptic signaling between electrically excitable cells. Electrically excitable cells means cell which has their potential is changed, and here the signaling is rapid, as we had seen in the endocrine signaling, that is the sorry, not in endocrine signaling, in the paracrine signaling, that is the signaling was very fast. Similarly, here in the ion channel, the signaling is rapid. So here mostly neurotransmitters, again the acetylcholine example can be taken for understanding the ion channel link signaling. In the image, as you can see that the channel is first closed, but when the specific ions bind to the channel, suppose we take an example of calcium. So if it is a calcium channel, when the calcium comes and bind to the ligands or the gates present on the cell membrane, that is the receptors, the channel will open and the molecules will be released from their higher concentration to lower concentration, whether it can be taken inside the cell or released from the cell, depending on what is the use or where the requirement of this ions are. So this is the second type of signaling. First we had studied about G protein coupled receptors signaling, second ion channel link signaling, and the third type of signaling is enzyme link signaling. So in the enzyme link signaling, this is different different than ion link signaling and similar to your G protein coupled receptor signaling. So this enzyme are mostly single pass transmembrane proteins. As we can see in G protein, there are 7 pass. So that is here the proteins was passing 7 times from the transmembrane. Here in the single pass, only one domain is there which is passing only one type from the transmembrane, or that is your cell membrane. So in this type of signaling, ligand molecule is always, ligand binding site is always outside the cell where the signaling molecule can come and bind, and whereas catalytic site or the effector site which bring about catalytic reaction is present inside the cell. So here as you can see, catalytic site is enzyme and they can directly work as enzyme, or they can indirectly be activated by binding to enzyme. So enzyme link signaling works in two mechanisms or two ways. That is, they can be directly, enzymes can be present as receptors and can be activated by the extracellular molecule, or they can be associated with some kind of receptors like G protein coupled receptors and get activated when the signaling molecules goes and bind to the receptor. So let us see how this works. So there's a first example where you can see the signaling molecule is usually in the form of dimer, and your receptor, inactive receptor, is present where the activity site, that is catalytic domain, is present inside the cell and receptor binding site is present outside the cell. So when the signaling molecules come and bind to the receptor, you can see the receptor get dimerized and your catalytic site is activated and it can bring about cell response. Whereas there's a second case where you can see that there's some kind of receptor to which the enzyme is associated or binded. So when the signaling molecules bind to the receptor, dimerization of the receptor takes place and the enzyme associated with that receptor will be activated. So there are two forms of enzyme activation: direct activation and indirect activation. And further after the activation, there can be cascade of cell signaling taking place to bring about cell signaling. So there are three forms of cell signaling based on what receptors are used: G protein coupled receptors, then second was ion channel link, and third is enzyme link signaling. So I think now you are clear about how the receptors work for cell signaling. Now we'll see an example in detail where we'll see that there are different molecules working together, that is three types of receptors: G protein coupled receptors, ligand or the ion channel receptors, and your enzyme channel receptors working together to bring about cell response. So let us discuss this example in detail. If you can see in the first point where the red color signaling molecule is there. So what happens? This red color signaling molecule goes and bind to the 7 transmembrane G protein coupled receptors which are in inactivated form. So when this signaling molecules goes and bind to the GPCRs, what will happen is GPCRs will be activated. That is, GDP will be converted to GDP, and the signaling molecule will bind to the receptors. And after binding to the receptors, when GTP is formed, this GTP will be separated and it will go and bind to some other protein. So in this case, it is going and binding to an enzyme known as activated phospholipase C beta. So further, earlier it was in activated form, it was phospholipase C beta. When it binds to the one subunit of your GDP or your GPCRs, it will get activated. And this phospholipase C beta is mostly hydrolyzing enzyme which converts pi 4,5 bisphosphate into diacylglycerol and inosyl triphosphate. So these are two molecules formed from pi 4,5 bisphosphate. That is, activated phospholipase cleaves pi 4,5 bisphosphate into diacylglycerol and inosyl triphosphate. And this diacylglycerol will further go and activate protein kinases, which bring about different types of cellular responses. Whereas inosine triphosphate, what it does, it goes and bind to the ion gated channels. So here the ion gated channels are the channels which are working for release of calcium in our body. So as you can see that, or you know that calcium is present inside the lumen of endoplasmic reticulum. So here when the triphosphate IP3 goes and bind to the receptor present on the, that is the ion gated receptor present on the lumen of endoplasmic reticulum, the receptor will get activated, that is the receptor will open or the channel will open and the calcium molecules will be released. So why this calcium molecule has to be released? So you already know that calcium molecules are required for muscle contraction and relaxation. So when this process has to be done, there is a signaling received from the outside of the cell, that is one cell sends a signal to the another cell, and this type of mechanism is activated to release the calcium. And further, we have already seen here that calcium goes and activated again protein kinase, and even DAG, that is diacylglycerol, also activates protein kinase, which can bring about some another type of cell response. So this is the best example where we can relate how all the 3 types of ligands, that is your or receptors, that is your G protein coupled receptors, ion channel receptors, and your enzyme link receptors can work to bring about different mechanism in our body. So this image I have taken from Cell, De Bruce and Alberts. You can refer further for detailed study. So let us summarize what we have discussed today. So first we had discussed what is cell signaling. So what is cell signaling? Signaling is a process required for communication between the two cells where the some signaling molecules work to bring about different types of cell signaling. So based on where the signaling molecule goes and bind to the far away site or to near away site, there are different types of signaling or molecules. That is autocrine signaling. Here the signaling molecules goes and bind to the receptors present on the cell itself. Second is paracrine signaling, where the receptors or the cell signals released from the cell goes and bind to the signaling molecules, goes and bind to the cells which are present in the vicinity, that is the neighboring cells. And endocrine signals are the signals which goes and bind to the cell which are present far away from the cell. And the second type of signaling here is based on the receptor mediated. So in the earlier type where different molecules were used, we had seen that the signaling molecule has to go and bind to the receptor present on different cells for activation. So there are 3 types of receptors. First is G protein coupled receptors, which has G protein which is activated by binding to the extracellular signaling molecules and then bring about cell response. Another is ion channel receptors, which also gets activated by binding to different types of ions, or different proteins can also activate this ion channel. And the last type is enzyme link receptors, which are activated by different receptors, the signaling molecules, where the enzymes get activated in 2 forms. That is, receptor itself can be an enzyme, or the enzyme can be coupled to any different kind of receptor. So this is the basic of cell signaling. So there's an assignment for you. I need all of you to discuss about how explain different forms of signaling, then explain or write note on GPCRs based on what type of structure G proteins have and what type of signalings they bring about, and lastly explain enzyme link signaling with examples. So this was all about today's session. In the next session, we'll try to link after when we have already seen the earlier session about cell transport. Then we have discussed about cell signaling, how signaling takes place. And now if when the signal has already released to the cell and if all the cell cycle of the cell has completed, cell will further go into cell death. So how this cell death takes place, whether it is due to aging or different factors bring about cell death, that is apoptosis or necrosis. So all this type of cell death we'll study in the next lecture. So thank you. So with this, I hope you have understood the basics insights into cell signaling.